Hello, today we're going to be talking about blood. Blood is the only fluid tissue within the entire body. It's a specialized connective tissue that is actually going to be consisting of formed elements. Formed elements consist of three things. When you take formed elements consist of three things. What are these three things that are a part of formed elements? These three things, which we're going to go into great detail of, are red blood cells, which also is known scientifically as erythrocytes. Then there's actually going to be white blood cells, which are leuco. Leuco means white and cytes means cells. And then there's also going to be platelets. Platelets means thrombocytes. Each one of these formed elements, they are believed to be living cells, except truly we can only say the only true living cell is the white blood cell. Because in order to be a true cell, a living cell, you need to have a nucleus, you need to have multiple amounts of organelles. And the white blood cell is the only one that contains two things. One of them is a nucleus, and the other one is organelles. This one, the red blood cells, have no nucleus. They are anucleated, and they have no mitochondria. Same thing with the platelets. They are anucleated. These are anucleated. The only true living cell is a white blood cell, but these are formed elements. That's one of the components of blood. Blood is actually, like I said, formed elements, which is made up of red blood cells, white blood cells, okay? And it's suspended, it's actually mixed in with a non-living fluid matrix called plasma. Plasma is actually going to be various amounts of solutes and water and protein and electrolytes and different kinds of nutrients and metabolic waste products, multiple amounts of things, hormones, and antibacterial enzymes, metabolic enzymes, respiratory gases, all these different kinds of things. Even toxins can be found within the blood. There's multiple things found within the actual blood plasma, and we'll go over to that in great detail when we get there. Just trying to introduce the subjects, though. But when we talk about blood, we usually categorize it in what we call a hematocrit. What is a hematocrit? A hematocrit is a test in which we take a test tube, and the first thing we do within the test tube is we line the test tube with an anticoagulant. What is the name of this anticoagulant? It's called heparin. Heparin is a natural anticoagulant that is actually going to help to inhibit thrombin, which is a clot-forming enzyme, by enhancing the activity of antithrombin 3. So what does heparin do? He enhances the activity of antithrombin 3, which inhibits the activity of thrombin, which is a very potent enzyme in helping to initiate clot formation. So he just inhibits any clots. Now, when you look at a hematocrit, they use a centrifuge, and they use a centrifugal force to separate the blood into three layers. When you see it, you'll see it looking like this. There'll be one layer, and this layer is actually going to be the most dense component, because it's at the bottom. The most dense things are at the end of the heaviest things are going to sink down to the bottom, so this is the most dense component. And when they calculated it, they calculated that this one accounted for, let's do this with a different color. This one is called, this is consisting of red blood cells. About 45% they calculated. It's the most dense component. So you'll see this one at the bottom. So normal, healthy individuals will have in their hematocrit layer, there at the bottom will be the most dense component they'll have, red blood cells, and it should account for 45%. People who suffer from, normal red blood cell amount is about approximately 4 to 6 million red blood cells, on average. There's about 4 to 6 million red blood cells. 10 to the 6. So we can say 10 to the 6 millimeter cubed of blood for red blood cells. Now normally what happens is sometimes the red blood cell, 4 to 6 million red blood cells. Sometimes what happens is if some people do what's called blood doping, it's a process in which they actually take some of their blood out of their circulatory system And when they take some of the blood out of their circulatory system, they put it in an area which has, right around 4 degrees C in a certain temperature in a certain room area, and they put it back into their circulatory system about a day or two before the event so that they can hydrate themselves so they have a good number of red blood cells. And that condition in which there is an actual red blood cell layer greater than 45% is called polycythemia. Polycythemia is an abnormal excess of red blood cells. Abnormal excess of red blood cells. Usually, there's different kinds of polycythemia. We usually say there's two kinds. There's actually what's called polycythemia vera. Polycythemia vera is actually a condition which is due to bone marrow cancer. A cancer, neoplastic cancer that's developing within the actual red bone marrow and is causing an abnormal proliferation of red blood cells. And that's a very dangerous condition. That usually you can check the blood, and usually if it's about 8 to 11, I believe it's about right around 8 to 11 million red blood cells, usually you're able to tell that they possibly might have polycythemia vera. And then there's what's called, and that would be a result of bone marrow cancer, again, polycythemia vera. There's secondary polycythemia, which is actually due to a condition in which sometimes it just can be due to enhanced EPO production, living in high altitudes, living in areas where there's less availability of oxygen, anything that increases the demand of the tissues for oxygen. Maybe you suffer from a severe hemorrhage and lost blood, anything that can actually decrease normal oxygen carrying capacities. But that's called secondary polycythemia, and that one only increases to about 6 to 8, so not much larger. Now, above that, there's a thin whitish layer, but we're going to draw it green. We're going to draw it green. So right here, again, what is the red blood cell layer? The red blood cell layer is the most dense component. It accounts for 45%, which is about 10 to the 6 millimeter cubed of blood, which is about, on average, if you want to remember the exact number, you want to be more than an average student, it's about 4 to 6 million red blood cells. Then, right around this erythrocyte, and this one's going to be called the plasma, right in here in between this on the erythrocyte plasma junction is called the Buffy coat. This is called the Buffy coat. The Buffy coat is it's accounting for about less than 1%. What is it? Less than 1%. What does this consist of? It consists of two things. Very, very important things. We're going to draw big. One is white blood cells. This is very important, white blood cells. White blood cells. And then there's another one called platelets. Another one called platelets. So when we think about the first one, again, let's go over this again. First one is the red blood cell layer. The red blood cell layer is going to be about 45%. Most dense component, that's right at the bottom. It's going to consist on average of about 4 to 6 million red blood cells in a normal human body. If it's elevated, it's called polycythemia. If it's above 45%, and that can be due to blood doping, it could be due to bone marrow cancer, it could be due to enhanced EPO production. And we'll go into the other one later. It's called anemia if it's decreased below 45%, below 4 to 6 million red blood cells. And then there's the Buffy coat, which accounts for less than 1%. That's consisting of platelets, also known as thrombocytes, and white blood cells, which is also known as leukocytes. About platelets it's right around, we say, usually 150 to 250,000 on average. Usually about 10 to the 5 millimeter cubed of blood. And then for white blood cells, we usually say around 4,800 to about 10,800 per millimeter cubed of blood. So which is about 10 to the, this is 10 to the 3. Because if you take the difference between the two, 10,800 minus 4,800 is about 6,000. 3 zeros, 10 to the 3. This one is about 100, so that's 100,000. That's going to be right around the area of 10 to the 5. So that's normally what we go with. We normally go with 10 to the 5 if you want to make it easier, 10 to the 6 millimeter cubed of blood if you want to just be an average student. And remember that the red blood cell layer, 45%, right around 10 to the 6 millimeter cubed of blood. If you want to be a really good student, you'd say 4 to 6 million red blood cells in an average human being. Buffy coat consists of less than 1%, which consists of white blood cells and platelets. Normally white blood cells are about 4,000 to 10,800 millimeter cubed of blood, and then we can say, if you want to just be an average student, about 10 to 3, because that just averages out millimeter cubed of blood. Platelets is about 150,000 to about 250,000 millimeter cubed blood, but if you just want to be an average student, you can say 10 to the 5. Then, above that, above this one, above the buffy coat, is actually going to be the largest layer. You know this layer? It's going to be more of a sticky amber fluid, which is going to consisting of various amounts of proteins and water and solutes and multiple other different kinds of things. You know this layer is called the plasma. This is called the plasma. You know the plasma consists of about 55%. So we say plasma accounts for about 55%. So when we normally go with all of this, we can say that the plasma is about 55%. That consists of various amounts of solutes, which, like I said, we'll go into great detail of later after we get done with this. This is just establishing the basic concepts of what is called a, what is this again? This is called a hematocrit. Okay, and then we'll go into all the details of what happens if you have deficient numbers of white blood cells, deficient amounts of platelets. And we didn't really go over deficient amount of red blood cells. What happens if you have excessive amounts of white blood cells, excessive amounts of platelets. Plasma, we're not going to go there. But we'll go over that in just a little bit. Actually, you know, we'll go over it now, just since we're on the same subject. But now let's just establish that we understand the hematocrit. Remember, what is blood? It's the only fluid tissue in the entire body. It's a specialized connective tissue consisting of a non-living fluid matrix called plasma, which accounts for 55%. It consists of various amounts of solutes and proteins and water. We'll go into detail about. It also consists of formed elements, which is 3 things. Red blood cells, which is also known as erythrocytes, white blood cells, which is also known as leukocytes, and platelets, which is also known as thrombocytes. We can say that when we do a hematocrit, we line the test tube with an anticoagulant called heparin, which enhances the activity of antithrombin 3, which suppresses the activity of thrombin, which is a clot-forming enzyme. If we do the centrifuge and it separates the blood into 3 components, those 3 components we can identify. How can we identify them? The most dense component, which is accounting for about 45% of the 100% of the hematocrit, it's consisting of red blood cells. It's the most dense component, so it'd be at the bottom. We say that the red blood cells in a normal human being not suffering from anemia, not suffering from polycythemia, a good student would know that there's about 4 to 6 million red blood cells. An average student would say 10 to the 6 millimeter cubed of blood. Okay? And then we have the buffy coat. You know, the buffy coat is actually going to be a thin, whitish layer right in between the actual, squished in between the plasma and the erythrocyte layer. It's about less than 1% consisting of white blood cells and consisting of platelets. And we're saying that there's no, you're not fighting off an infection. You're not actually you're not suffering from leukocytosis or leukopenia. You're not suffering from thrombocytopenia or thrombocytosis. You're not suffering from any of those kinds of conditions. We're just talking in general. A normal human being, less than 1%. The white blood cell, if you're a good student, you'd say 4,800 to 10,800 white blood cells in a per millimeter cubed of blood. If you just want to be an average, you can just say 10 to the 3. Platelets, about 150,000 to about 250,000 platelets in a millimeter cubed of blood, or you can just say 10 to the 5 platelets per millimeter cubed of blood. Then we go into the least dense component, which is actually going to be the plasma, which is actually going to be about 55%. We'll go into great detail about what is in the plasma. Okay, let's really quickly go into what is, how can you identify, what is it if there's above 45%? What is it if it's below 45%? Yada, 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 all these great things. Okay, let's go over on the top one over here. This one was white blood cells, so we'll just go down. You know, white blood cells, normally, like I said, we can normally say that white blood cells are actually going to be right around the area, we said right around 4,800 to about 10,800 millimeter cubed of blood. We can say. Now, you know, if someone suffers from an infection, or they suffer from an injury, a tissue injury that maybe came from maybe bacteria, or that came from a fungal infection, or it came from a parasitic infection, or sometimes even in response to burns. When somebody actually gets burned, it depresses the immune system. But any kind of immune response, you know, that would cause the bone marrow to be able to enhance the process of increasing the actual, what's called leukopoiesis. We'll go into that later on when we go into the bone marrow, and we'll talk in that in great detail. There's what's called leukopoiesis, and what happens is in response to an infection or an immune response. You know, there's different kinds of chemicals like interleukin-6 and granulocytic colony stimulating factors and agranulocytic colony stimulating factors, different kinds of growth factors which can stimulate the process of leukopoiesis, increasing the white blood cell formation. And in response to that, that increases the white blood cell amount. So if the white blood cell amount is beyond 4,800 to 10,800, we say if it's beyond that, if it's above, so let's break this into two categories because it only can be two. If it's above the normal amount, above normal, that is called leukocytosis, which is a increased number of white blood cells. We say normal response is right around 11,000. We say a normal response to an infection is about 11,000 white blood cells per millimeter cubed of blood. If it is below normal, we call it something else. We call it leukopenia. Now, leukopenia is a little bit different. Usually leukopenia is in response to cancer. What happens in cancer is obviously it's going to increase your white blood cell formation because your white blood cells are going to want to be able to destroy the different kinds of immune system cells. But what happens is when they do the cancer, they give what's called anti-cancer drugs. Anti-cancer drugs, depending upon whatever the cancer is. And those anti-cancer drugs can help to destroy some of our actual white blood cells. And sometimes that can actually cause leukopenia. Anti-cancer drugs in response to fighting off cancer. And there's another one that some of you might be familiar with. It's called excessive amounts of cortisol. You know, cortisol is actually going to be a glucocorticoid, which we'll learn in chapter 16, the endocrine system. Cortisol is a glucocorticoid that has so many functions. It helps to be able to promote what's called gluconeogenesis, the formation of a glucose molecule from non-carbohydrate sources like amino acids that need to be deaminated and oxidized and so on and so forth, or glycerol. It also helps to be able to increase the sensitivity of a neurotransmitter that is released by the sympathetic postganglionic motor neurons called norepinephrine, or released by the adrenal medulla norepinephrine, which increases blood pressure. It can also help to be able to cause the catabolism of proteins in our tissues, our cellular tissues, and that can cause that. It can also initiate what's called lipolysis, breaking down triglycerides or triacylglycerols within the adipose tissue, and sometimes even within the liver. It even is very helpful in stressful situations. There's receptors on the hippocampus that respond to cortisol, which affects memory. And you know, there's multiple other things that cortisol can do, but one of the main ones, and the whole purpose of me mentioning this, I got off track, but cortisol inhibits inflammation. That is one of its very, very powerful and potent effects. But sometimes people can receive too much cortisol. And when they receive too much cortisol, the whole job of cortisol is it binds onto receptors that inhibits inflammation. You know, like the release of histamines, it inhibits the release of bradykinins, and inhibits the release of different kinds of like prostaglandins and leukotriene and all these many kinds of agents that help to be able to attract an immune system cells. And by doing that, it can depress the immune system. And so it can actually inhibit any kinds of immune system cells coming to fight off infection, even though there could be a very dangerous fungi or parasite or pathogen or infectious agent within the area of a tissue. But just remember that white blood cells can be broken down into the important idea is just normal levels. Let's make sure we establish that. This is normal. You know, normal. Who's normal anymore? This is normal levels. Normal levels is 4,800 to 10,800 white blood cells per millimeter cubed of blood above normal is called leukocytosis. That's usually in response to, everybody usually knows that, that's in response to usually a infection or of some sort that is causing, sometimes it can even be more than that, but usually it's in response to an infection. And then below normal is usually called leukopenia. Leukopenia is usually due to if someone has cancer, they usually are receiving anti-cancer drugs, and these anti-cancer drugs are destroying the white blood cells, and that can actually decrease white blood cell production. And sometimes people receive in response to maybe inhibiting inflammation, or whatever it might be, cortisol, epidural injections, too much cortisol, or pharmaceutical doses that are too large that inhibit inflammation, but in response undesirably depress the immune system. So that's what we would say if there was a decreased or increased level of white blood cells. And there's different kinds of diseases that describe all these white blood cells. You know, there's multiple more than just leukocytosis. There's leukemia, there's infectious mononucleosis, the Epstein-Barr virus, there's the Hodgkin's, non-Hodgkin's lymphomas, and just multiple different kinds of diseases that we might get into, I don't know, maybe later in future videos. But now let's go into red blood cells so we can actually get into the real good stuff, and that's talking about how red blood cells are made, going over their lifespan and their function, and multiple other things. Now let's go over red, that's ugly R, red blood cells. We say that a normal level of red blood cells, normal levels are usually approximately, like we said, about 4 to 6 million red blood cells per millimeter cubed of blood. Now, if someone is suffering, we've already talked about one, so we can cancel it out. I'll review it just briefly. We've already talked about it. If someone has a condition in which it is above normal,